Hello viewers, in previous video I explained about the isolation of protoplast from leaf tissue. Now in this video I am going to explain about the isolation of protoplast from cell suspension cultures and also the culturing process of protoplast. First we'll see the isolation of protoplast from cell suspension cultures. Here, the cell suspension cultures are considered as the most suitable material for the protoplast isolation. More number of protoplast can be obtained only after subculturing the new cell suspensions. Fresh cell suspensions preparations avoid all the obstacles and end in successful isolation. In this picture, you can see the isolation of individual cells or single cells from the cell suspension. In one way, the, the individual cells containing test tube take and centrifuge in centrifugation. Then after centrifugation, the precipitate, individual cell precipitate is settled at the bottom. From that settled single cells are isolated by using pipette. And in another way, here then next after isolation of single cells from the cell suspension, we have to do the isolation of protoplast. Now we'll see the procedure of isolation of protoplast. Here several steps are involved in the isolation of protoplast through the cell suspension cultures. First, we have to do the filtration of harvested cell suspension through a nylon sieve. And then after filtration, the filtrate contains single cells and cell clumps. Then we have to do the separation of the cell suspension medium and single cells. Then after separation of single cells and medium, we have to transfer these single cells into a flask. Then we have to add the plasmolyticum to the flask which contains single cell. Here the plasmolyticum is cell and protoplast washing medium, that is CPW, and 13% mannitol solution. Then these cells are subjected to the plasmolysis. After adding the plasmolyticum to the single cells in a flask, it leads to the plasmolysis of the cells. Then after plasmolysis, we have to do the separation of plasmolyticum and cells. Then after separation of plasmolyticum and cells, we have to add the enzyme solution to the cells into the flask. And after adding the enzyme solution, we have to do the rotation of the flask with a rotatory shaker for 4 to 6 hours. Then after rotation on the rotatory shaker, after 4 to 6 hours, then we have to do the separation of these enzyme solution and cells by the filtration. And after filtration, we have to do the transforming the filtrate to a centrifuge tube which contains the single cells. Then after centrifugation, collect the sediment which contains the protoplast pellets. And after removal of supernatant enzyme medium, resuspension of protoplast pellets in CPW, cell and protoplast washing medium, and 2% sucrose solution. And after resuspension, we have to subject it to spinning. And after spinning, collect the viable and pure protoplast at the surface of the test tube, then washed with CPW, cell protoplast washing medium, and with 13% mannitol with repeated centrifugation. And the suspension of protoplast in measured volumes of liquid culture medium in the final step. Then after isolation of protoplast either from leaf tissue or from cell suspension culture, the next second major step in the culturing process of protoplast is culture of protoplast, the culturing of isolated protoplast. Okay, here uh, isolated protoplast can be cultured in several ways or in several methods, of which the agar embedding technique is a common method and widely used method for culturing of protoplast. In this technique, the protoplast suspension is mixed with equal volume of melted agarified medium. Then the protoplast agar mixture is poured into a small petri dishes. Then this embedding of protoplast in agar medium is called plating, and this mixture is allowed to spread uniformly. After replacing the lid on the petri dish, the petri dish is inverted and incubated at 25 degrees Celsius with illumination. And the culture are subcultured periodically in the same agar medium. And the developmental stages of embedded protoplast can be studied under the compound microscope. The protoplasts are gradually developed the cell walls and become regenerated cells. Later, these cells divide and redivide and form colonies. Cell colonies are a group of cells. These colonies are subcultured on agar medium to produce new cultures. Here, during the culturing process on agar medium, shoots and roots are induced on the finally developed callus. Okay, this is about agar embedding technique. In this picture, you can clearly see the steps which are involved in the agar embedding technique of culture of protoplast. Okay, in the first we have to take the isolated protoplast in a test tube, then we have to pour in a petri dish and allow the spreading uniformly throughout the petri dish. Then we have to invert the petri dish. After inversion of petri dish, transfer in incubation culture room and incubate at 25 degrees Celsius with illumination. Then the after serial subcultures, the protoplast in the medium gradually develops the cell walls and become regenerated cells. And in some weeks, after some weeks, these cells are divided and redivided and form cell colonies. These colonies are subcultured on agar medium in a petri plate. These agar medium during the culturing process, shoots and roots are initiated. Then after root initiation, we have to transfer into pots for further growth and development of plants. Okay, besides this method, the culture of protoplast can also be carried out by several other methods like droplet culture, co-culture, feeder layer, Hanning droplet method, and bead culture. But most widely used culture technique of protoplast culture is the agar embedding technique. Thank you, guys.